Hello there! Welcome to Part 3 of plasma membrane and its role in drug absorption. In Part 1, we covered the structure and features of the plasma membrane. In Part 2, we looked into transcellular transport, its types, and its features. Now, we are moving to the next important topic, paracellular transport. When we consider how the body moves substances across tissue barriers, we often think of cells as solid walls. But there's a critical pathway that avoids the cell entirely. This is paracellular transport, distinguished from transcellular transport. Paracellular movement occurs between adjacent epithelial or endothelial cells through intercellular clefts without crossing cell membranes. In contrast, transcellular transport involves substances moving directly through cell membranes. Paracellular transport is predominantly passive, relying on gradients rather than cellular energy. The structural basis of this route starts with the intercellular space. These narrow extracellular gaps are filled with fluid and serve as specialized aqueous pathways. Because they are water-filled, they are ideal for small hydrophilic molecules that cannot pass through fatty cell membranes. In clinical practice, this pathway is crucial for transporting key molecules like glucose, urea, and various water-soluble drugs. For example, glucose and urea are efficiently absorbed via the paracellular route in the kidneys and intestines, while certain small drug molecules are designed to use this passageway for effective absorption. The master controller of this space is the tight junction, or zonula occludens. Located at the apical region of cells, these junctions form a seal that regulates the permeability of the barrier. This seal is made of a complex protein machinery, claudines. These are the most important. They form the selective pores that decide which ions get through. Occludines. These manage the integrity and regulation of the barrier. Junctional adhesion molecules, JAMs. These act like the glue, ensuring cell-to-cell adhesion. They handle cell signaling and play an essential role in guiding white blood cells, leukocytes, between cells during migration. This function becomes especially clinically relevant during inflammation or infection. Zonula occludens proteins 1, 2, and 3. These provide the internal scaffolding, anchoring the junction to the cell's own skeleton. We often think of tight junctions as just biological glue, but they are actually dynamic gatekeepers with 3 distinct roles. First, they provide the barrier function. This prevents the unrestricted passage of substances and maintains the tissue's electrical resistance. Second, they act as a fence. This fence separates the apical membrane at the top from the basolateral membrane at the bottom, ensuring that the cell keeps its up and down polarity. When it comes to actual transport, there are 2 distinct modes. The first is the pore pathway. Picture the pore pathway as a selective checkpoint. It restricts entry based on size and charge, allowing only certain ions and small solutes to pass while blocking others. Unlike the pores, the leak pathway is non-selective. It allows larger molecules within a limited range to pass. This is not controlled by claudines, but is regulated by occludin and the cell's cytoskeleton. It is a broader route that handles the overflow of larger solutes. The leak pathway becomes especially important under certain physiological and pathological conditions. For example, it is more active in tissues such as the renal proximal tubule, where bulk reabsorption is needed. It is also upregulated during inflammation or injury when the barrier becomes more permissive to immune cell access or cytokine signaling. However, increased activity of the leak pathway can also lead to unwanted leakage and contribute to disease when barrier integrity is compromised. There are 4 primary engines driving this movement. The concentration gradient. This is simple diffusion. Solutes naturally move from areas of high concentration to low concentration. 2. The electrochemical gradient. This is crucial for ions. It's the combined pull of electrical charges and chemical density. The osmotic gradient. Here, water is the driver. As water chases higher solute concentrations, it creates a solvent drag, pulling other small molecules along. The hydrostatic pressure gradient. This is a physical push of fluid across membranes, a process vital in our capillaries and the kidney's glomerulus. There are three core features you need to remember about this pathway. First, it is governed by permeability. The looseness, or tightness, of these junctions determines how much can get through. Second, it is passive. Unlike active transport, which requires ATP or cellular energy, paracellular movement has no metabolic cost. Particles move freely along gradients. Third, it is selective. While we used to think of these as leaky holes, we now know they are sophisticated. They filter based on charge and size, a process managed by a family of proteins called claudines. Next, moving to the different types of paracellular transport. The most common way substances move is through passive diffusion. This is governed by Fick's law. Essentially, solutes move from high concentration to low concentration. It's a game of size and charge. Whether it's sodium, chloride, or urea, the tight junction acts like a selective filter. If the molecule is the right size and carries the preferred charge, it slips through the aqueous pores between the cells. Then, there is solvent drag, also known as bulk flow. Here, the solutes aren't moving on their own. Instead, water moves across the barrier due to osmotic or hydrostatic pressure, and it creates a drag force. It's like a river. As water flows, it carries electrolytes and glucose along. This process is vital for absorption in the small intestine and kidney reabsorption. Closely related is paracellular convection. Driven by pressure gradients, this is less about individual molecules and more about the simultaneous movement of multiple solutes. You'll see this most prominently in the glomerular capillaries of the kidney, where filtration happens at high volumes. Finally, the system gets incredibly specific through ion-selective transport. Different types of claudines create specialized VIP lanes for specific ions. Claudin 2 forms a cation-selective channel for sodium. Claudin 10 prefers anions. Claudins 16 and 19 work together to move magnesium and calcium. Claudins are not static structures. They can change their shape or conformation, and this dynamic behavior allows them to regulate and facilitate the selective movement of ions and small molecules through tight junctions. It is this precise molecular engineering that allows our organs to maintain the perfect chemical balance required for life. If the tight junction is a gate, then the proteins are the operators. While claudins and occludins form the physical barrier, a group called ZO proteins acts as the scaffold, anchoring everything to the cell's internal skeleton. Together, they act like a biological rheostat, adjusting the pore size, ion selectivity, and overall permeability of the tissue in response to the body's immediate needs. Not all barriers are created equal. There are two distinct epithelial builds: leaky epithelium and tight epithelium. These differ fundamentally in structure, function, and permeability, depending on tissue needs. The leaky epithelium, found in places like the small intestine and the proximal renal tubule. These have loose junctions designed for high-volume transport. They have low electrical resistance, allowing for massive solvent drag to move nutrients and water quickly. The tight epithelium, this is the high-security version. You'll find this in the distal nephron of the kidney and the blood-brain barrier. These have extremely high electrical resistance and minimal paracellular leak, ensuring that only the most strictly invited molecules pass. Even with the gates open, molecules don't move without a push. There are three primary forces driving this transport. These are concepts we've already heard before, but now we're seeing how they specifically apply to paracellular transport. First, the electrochemical gradient. This is pure physics. Electrochemical gradient. Ions move toward opposite charges, like Na+ toward negative areas. Next, the osmotic gradient. Think of this as the sponge effect. It creates what we discussed earlier, solvent drag, pulling small solutes along with it. Finally, hydrostatic pressure. This is a mechanical force, like a piston. Hydrostatic pressure, mechanical push, like a piston, drives fluid and solutes across capillaries. Now, you can clearly see how they work together to drive movement through the paracellular pathway. To see these mechanisms in action, look no further than your gut. Intestinal absorption is a masterclass in teamwork. It starts with transcellular transport, where the cell actively pumps sodium across its membrane. This creates a powerful osmotic gradient. Nature hates an imbalance, so water rushes through the paracellular gaps to even things out. As that water moves, it creates the solvent drag we discussed, pulling essential electrolytes along with it. This is the primary way your body stays hydrated and nourished. In the kidneys, paracellular transport is a heavy hitter. In the proximal tubule and the thick ascending limb, massive amounts of filtered material need to be reclaimed by the body. Through these cellular gaps, the kidney reabsorbs a constant stream of sodium, chloride, calcium, and magnesium. Without this high-volume backdoor for ions, our renal system would be far less efficient at maintaining our internal mineral balance. At the level of the blood vessels, we see a different application. In our capillaries, fluid and solutes are exchanged through intercellular clefts. This allows for rapid delivery of nutrients to tissues and the removal of waste products. However, in the placenta, the rules change. Here, paracellular movement is intentionally limited. Only the smallest solutes can pass through the gaps, protecting the developing fetus by ensuring that most transport is strictly regulated by the cells themselves. Clinically, this regulation is vital. If the paracellular barrier becomes abnormally permeable, as seen in conditions such as preeclampsia or certain infections, harmful substances or immune cells may cross more readily, potentially compromising fetal health and leading to complications. Now let's see how the body regulates this dynamic barrier in real time. The paracellular barrier isn't a static wall. It's a living, breathing system that responds to molecular signals in real time. This is achieved through two primary methods. Claudin expression. The cell can literally swap out the bricks in its wall. By changing which Claudin isoforms are expressed, the tissue can become more or less permeable to specific ions. 2. Phosphorylation. Think of this as a chemical toggle switch. When certain enzymes add a phosphate group to tight junction proteins, they can trigger the proteins to tighten or relax their grip, instantly altering the barrier's strength. Your hormones act as the body's long-distance remote control for these gates. They can tighten or loosen the junctions depending on the physiological state. Aldosterone. Known for its role in salt balance, it indirectly influences paracellular ion movement to ensure the body retains the right amount of sodium. Cytokines. These are the signaling molecules of the immune system. While necessary for defense, they often signal the junctions to loosen up, allowing immune cells and fluid to reach an area of need. However, this regulation can be hijacked or disrupted. In pathological regulation, the barrier's integrity is compromised. Inflammation. Chronic inflammation can lead to the persistent breakdown of tight junction proteins. Infection. Certain bacteria and viruses specifically target these junctions to make the tissue leaky, allowing pathogens to bypass the body's natural defenses. This leaky barrier is a hallmark of many gastrointestinal and systemic diseases. Now let's take a look at the clinical relevance of paracellular transport. Understanding the paracellular path isn't just academic. It's the key to modern medicine. Take drug absorption, for example. Many hydrophilic or water-loving drugs rely on these gaps to enter the bloodstream. However, because tight junctions are so restrictive, large molecules often get blocked. To solve this, scientists are developing permeation enhancers and TJ modulators, chemical keys that temporarily relax the junctions to let life-saving treatments pass through. One of the greatest medical triumphs in history is oral rehydration therapy. In cases of severe dehydration, like during cholera, we use the paracellular mechanism to save lives. By giving a specific balance of salt and glucose, we trigger a massive uptake of sodium. This creates a powerful osmotic gradient and solvent drag, literally pulling water through the paracellular gaps and back into the body. It's a simple solution to a complex physiological problem. When these barriers fail, the results are often devastating. In inflammation and celiac disease, the epithelial barrier is disrupted. This leaky state allows unwanted substances to slip into the bloodstream, triggering further immune responses and diarrhea. In cholera, the infection causes excessive ion secretion into the gut. Water follows those ions out through the paracellular route, leading to the rapid, dangerous fluid loss we see in the disease. In the kidneys, paracellular transport plays a key role in the renal tubules. Here, important ions such as sodium, calcium, and magnesium are reabsorbed between cells rather than through them. This helps maintain electrolyte balance and supports overall fluid regulation in the body. Finally, we look at the blood-brain barrier. This is the Fort Knox of the human body. The junctions here are so extremely tight that they prevent almost all paracellular drug entry. While this protects the brain from toxins, it is the single biggest challenge in treating central nervous system diseases. Finding a way to safely unzip these gates for a few moments is the holy grail of neuropharmacology. We've seen how the gates are built and powered, but what actually determines the volume of traffic? It comes down to a few critical factors, starting with molecular size. In the paracellular world, size is everything. Smaller molecules, such as water and simple ions, can easily slip through tight junction pores. Larger molecules, such as proteins or complex drugs, find the path nearly impassable unless the junctions are specifically modified to allow them through. Next is charge. As we've seen with Claudins, these pores aren't just holes, they are electrically charged tunnels. Depending on the specific proteins present, a junction might act as a cation-selective path, welcoming positive ions, or an anion-selective one. But even the best filter fails without tight junction integrity. This is the single major determinant of transport. If the structural proteins are degraded by disease or chemical signals, the barrier loses its selectivity entirely, leading to uncontrolled leakage. Clinically, compromised tight junctions are at the core of diseases such as celiac disease, where inflammatory reactions disrupt the intestinal barrier, allowing antigens and toxins into the bloodstream. We also have to consider surface area. It's a simple ratio. The more surface area you have, the more junctions are available for transport. This is why the massive, folded internal surface of your small intestine is such an efficient engine for paracellular absorption. Finally, your physiological state changes the rules of the game. Your body doesn't transport nutrients the same way when you are fasting as when you are fed. In a fed state, the presence of nutrients like glucose can actually trigger the junctions to loosen to aid in absorption. And as we've seen, disease states can turn a finely tuned filter into a wide-open door. Paracellular transport has some clear advantages. It's completely passive, so it doesn't require any energy. This makes it an efficient pathway for the rapid movement of water and small solutes, playing a key role in maintaining fluid balance and electrolyte homeostasis. However, it also has limitations. Compared to the transcellular route, it offers less precise control over selectivity. Typically, if a molecule is larger than 500 Daltons, it's simply too big to fit, and is highly dependent on the integrity of tight junctions. Because of these constraints, it is not suitable for transporting large molecules like proteins or peptide-based drugs. Moving to the summary, paracellular transport is the body's passive bypass, moving substances between cells rather than through them. It's essential for fluid and electrolyte balance, and its efficiency depends on the dynamic permeability of tight junctions. Clinically, disturbances can lead to dehydration, edema, or electrolyte imbalances. This pathway underpins kidney filtration and oral rehydration, and even impacts drug delivery. Mastering it helps us understand and manage these critical physiological processes. And with that, we come to the end of our complete series on plasma membrane and its role in absorption. If this series helped you understand the concept of absorption better, don't forget to like, share, and subscribe.